Hi. In previous session, I have introduced about the immune organ, and I have explained there are two major classes of immune organ, that is primary and secondary lymphoid organ. The primary lymphoid organ basically the site for generation of immunocompetent lymphocytes. It could be B cells, it could be T cells. And another set of organ, we call it as a secondary lymphoid organ, and these secondary lymphoid organs are basically playing a very important role role in interaction between uh, immunocompetent lymphocyte and antigen, and over there is an appropriate environment. So these secondary lymphoid organs are very important. Okay, so now I will take you to various immune organ, and first we will take the thymus. So as you know, this is a primary lymphoid organ, and this is a key site for the development of T cells, and we call it as a thymocyte. Okay, so basically this thymus is you can see in this slide that the thymus is basically present over the heart, and it is basically having a uh, it is present over the heart, and there are several organs, as you can see in this slide. There over the diaphragm there is a heart, and over the heart there will be a thymus. Okay, and this the precise anatomical location is that it is anterior superior means above, mid sternum, bilobed organ on the top of heart. Okay, basically this thymus is a pinkish gray color soft tissue, and if you want to visualize this organ in mice, then you need to open the very young mice. I will tell you in a short while that this thymus is a unique organ which is bigger in size in early ages, and this start degenerated as the animal or human age. Okay, so this is a kind of unique property. It's not growing, it's a other way around. It's basically reducing in the size. Okay, so this is basically in human, in infant it is about 30 gram weight of this organ is 30 gram, and at the age of 35 its weight reduces to about 24 grams, and at the age of 65 its weight only 2 gram of this thymus tissue remains. Of course, there will be a lot of fatty tissues present in that organ, but the active organ will be only 2 grams. So you can understand this thymus is having a unique property. So with age it is reducing the size. Okay, and this is a place where T cells are generated, and it's very interesting to know that most of T cells which is generated in or educated in the thymus, most of these cells are basically undergo apoptosis, programmed cell death, or they die. Okay, and why they die? Because there is a very strict selection process in the thymus. Okay, in order to become a immunocompetent T cells, immunocompetent naive T cells. Okay, and basically this phenomena we call it as a clonal selection or clonal deletion. So this is a process by which the T cells finally select for the generation of immunocompetent naive T cells. Okay, and I will take this phenomena, this clonal selection and clonal deletion, later on when we will probably study the T cells. Okay, so at the moment just you can understand that this clonal selection and clonal deletion is there is a lot of cellular and molecular changes taking place in the cell, and all those cells which is reacting with our own antigen, they are basically eliminated, they are deleted, or they undergo the apoptosis, and all those cells which is not reacting with our own cells, they are selected. So this is in a brief, this is the meaning about the clonal selection and clonal deletion. Okay. Now let's look at the cross section of this thymus, how the thymus looks like. Basically, it has a two major region. One is a cortex region. Generally, this cortex region is the outer region of the organ. Okay. And there is a medulla region, which is inner side of the organ, and here you can see that there are dividing thymocytes, and this cortex region is basically quite densely packed, okay, with immature T cells, okay. And here you can see that there is a lot of dividing thymocytes, and there is a structure known as trabecula. This is a kind of partition. So there will be a subsection in the organ. Okay. So this trabecula is basically a separating the various section in thymus, and there is a capsule. It's a kind of covering over the thymus, and there are here you can see that there are dead cells. So all those cells which is autoreactive, they are programmed to die. Okay, they are basically dead cell, and there are some cells which is alive, and they are probably become a immunocompetent naive T cells. And in addition to this, there is a nurse cell which basically provides the factors which is needed for the growth of these cells. Okay. And there is a cortical epithelial cell which is again providing some or other factor in order to grow these thymocytes. And there are interdigitating this dendritic cells. So all these cells plays a very important role in development of these immunocompetent naive T cells. And there is a one structure which we call it as a Hassel corpuscle. So basically this provides basically eosinophilic type 4 reticular cell. This basically provide a cytokine molecule which we call it as a TSLP. It's a thymic stromal lymphopoietin, and it is needed for the development of these cells, these thymocyte, developing thymocyte. And here you can see that there are macrophages, dendritic cells, as I explained you previously. So all these cells basically support the development of T cells, okay, or thymocyte, or developing thymocytes. And it's very interesting. If there is there are several cases in which this thymus is not present in the baby, and those individuals, or if we do the experiment, if we remove the thymus in early stage in the mice, then there will be a severe reduction of T cells in that particular animal. Okay. And those animals are kind of quite immunocompromised. They are susceptible to many infection, and generally they cannot make a long life. Okay. So you can understand this thymus is very important in early stage of life. Okay. And there are some in human, there are some cases in which the thymus is absent. This is due to some congenital issues or some developmental issue, or it may be associated with genetic. So those individual, they cannot survive very long, and at early stage they will have a recurrent fever and a lot of recurrent infection, and those patient, we call it as a Di-George syndrome. Okay. So this you can understand the importance of thymus, and here I want to mention that at early stage you may have this question that, okay, this thymus is reducing with the age. So our immune system, how our immune system work at a later stage. So here I would like to say that these functions are taken over by the lot of mucosal associated lymphoid tissue. Okay. And we will discuss later, maybe in this session or next session, about the mucosal associated lymphoid tissue. So now another primary lymphoid organ is bone marrow. So bone marrow is a very important place for the generation of almost all cells which is present in the blood. Here you can see there is a bone marrow, and this bone marrow is basically the cavity which is present in long bones. Okay. So over there, this there is a one specialized cell which we call it as a hematopoietic stem cell. So this hematopoietic stem cell differentiate into variety of blood cells, as well as they are differentiate into the immune cells. In bone marrow, the most important cells which are generated are the B cells. Of course, besides other blood cells like platelets and another white blood cell, we will discuss all those immune cell such as neutrophil, basophil, eosinophil. All these cells are generated from this common progenitor, which we call it as a hematopoietic stem cell. So then they differentiate into various lineages, and then they make various kind of immune cells. Bone marrow is a key site for the development of or generation of the B cells. Okay, this is a site for generation of B cells besides other cells, and this bone marrow also kind of age with aging, means the most of this the bone marrow cavity is filled with fat cell as the individual age. Okay, and probably due to that there is a declination of immunity in those individual. Okay, so with the advancement of age, most of this bone marrow cavity is filled with fat cells. Okay, which is about 50%, and this is a site for B cell. And how B cells, here you can see that there are different stages of development of B cell. Here I am just showing a snapshot. I am not going to take this thing in more detail. I will discuss when I will take up the B cell development, but just for your information I want to show that okay, in bone marrow this B cells are started developing from hematopoietic stem cell, and when the B cell exit from bone marrow, they are mature immunocompetent B cells, and then they will move to the peripheral lymphoid organ, mainly spleen or some lymph node, and over there they will interact with antigen, and then they will become a means, and they will generate the antigen specific antibodies after differentiation to the plasma cell. Okay, so we will take up all those things in great detail when we will take the antibodies or B cell development. Okay, so bone marrow is a site for the development, key site for the development of B cells, but it is not true for other animal. For example, in birds, this is a there is a specialized organ which we call it as a bursa of Fabricius. This is an pouching of this intestine. Over there this B cells are developing. So basically this is common in the birds. Okay, I will show you. In cattle and sheep, the spleen is the key site for the development of B cells, and Peyer's patches are also the site for the development of B cells. In human also, later on, in later stage, when this bone marrow is not so functional or not so active, at that time this mucosal associated lymphoid organ are basically making the B cells. Okay, and in mucosal associated lymphoid tissues this Peyer's patch comes. Okay, in rabbit this is the appendix. Appendix is a site for the B cell development or generation of B cells. Okay, here this is a organ which is I was talking in birds. Here you can see that this is basically towards end. Okay, and this is taken out from the bird, basically the hen, and here you can see in this bird also this organ, this bursa of Fabricius is there. and this is the site for B cell development in avians, mainly avians, okay, birds. Next organ is the spleen, which is a secondary lymphoid organ. If you remember, the primary lymphoid organ is thymus and bone marrow, and secondary lymphoid organ or peripheral lymphoid organ is spleen and lymph node and mucosal associated lymphoid tissue. So, now we will discuss about the spleen. So, this is a location of a spleen. Here you can see the spleen is located in our body, and this spleen has a this is a cross section of a spleen, and here you can see there are two major regions. One is one we call it as a red pulp. You can see in this slide that red area is we call it as a red pulp, and there is a white area and we call it as a white pulp. So, red area is not so important in point of immunity. The white area is most important for the immunity. Okay. Here you can see various structure, and there is a capsule which covers this this organ. It's a kind of membranous kind of thing and consists of epithelial cell. Okay. And there is a trabecula which is just again making a partition in the spleen, and there is a vascular sinusoid. So, vascular sinusoid is a it's a kind of open structure. There is an over there blood is there, but it's there is no blood vessel. So, blood is flowing in a space. Okay, we call it as a vascular sinusoid, and all this area we call it as a red pulp. And when you look at the white region, there is a primary follicle. I will explain you what is primary follicle, and there is a marginal zone, and there is a structure which we call it as a periarteriolar lymphoid sheath. Okay, so this constitute the white pulp. And what is this primary follicle? So, here I will just give you the concept of primary follicle, secondary follicle or germinal center. So, primary follicle is the region in which this antigen is present, which is not yet reacted with the immunocompetent lymphocytes. Okay. So, over there is a basically immunocompetent naive lymphocytes are there. Another is there is a FDC. FDC stand for follicular dendritic cells. Okay, and there is a resting B cells. Resting B cells, or you can also understand is as a immunocompetent naive B cells or resting cells. They are same thing. Okay, so this primary follicle is consist of these component. And there is another term which we call it as a secondary follicle. Secondary follicle is over there will be a lot of interaction between FDCs, antigen presenting cells, dendritic cells, all are very active. Okay, they will be actively secreting the plasma, making or differentiating B cells are differentiating to the plasma cell. So, all those very active area we call it as a secondary follicle or germinal center. So, when the antigen will be will come, initially it will interact and there will be a no activity, and then we call it as a primary follicle, and later on when there is a lot of immune reaction is taking place, and then there will be a differentiation of this naive B cell into the this naive B cell will differentiate into the plasma cells, and that will actively secrete the antibody molecule. Okay, so those centers we call it as a germinal center. In experiment, we basically when we challenge the animal with some antigen, then we look for this germinal center. Okay, which is very active by immunohistology. Okay. So, this is the structure of the primary follicle, and probably you may understood about this germinal center or secondary follicle. So, this is a structure of spleen, and there is a here you can see there is a vein, artery and germinal center. So, germinal center, here you can see it's a much more compact and very active. Okay. So, what is the function of a spleen? So, the most important function of a spleen, which you have studied probably in earlier classes, this is a place where the quality control of blood cells are taking place, particularly RBCs. Okay. We also call it as a graveyard of RBCs. So, whatever RBCs which is old, aged, damaged, everything will be removed by the spleen. Okay. Basically, all blood will filter or pass through this organ, and whatever the damaged RBCs are there, they will be removed. Okay. So, this is a one of very important function. So, due to this function, the spleen is a place where all blood borne antigens are coming, and there will be a development of immune response. Blood borne means, so antigen can go by various ways. Okay. One way is that the antigen is transported in our blood, in blood vessel. Then this antigen will go into the spleen, and over there this antigen will be trapped, and then there will be a development of appropriate immune response. Okay. Appropriate immune response means there will be a development of B cell mediated immune response, T cell mediated immune response. Okay. So, the immunity point of view, this is a one of important function, and if non-immune function is the removal of damaged RBCs, and it will be removed, and then the blood will be again returned, okay, to the system. So, I already explained this trap the blood borne antigen just now, and if we remove the spleen from young individual, then that can cause a severe problem. It will cause a systemic infection, and among systemic infection, these some bacterial infection is very common, such as Streptococcus pneumoniae. Okay. So, this basically very invasive bacteria, and that can cause various wide range of symptoms. It can affect the central nervous system. Another is Neisseria meningitidis. This infection, basically infection of this bacteria, result to the some neurological problem. It basically affects the central nervous system and meninges. So, meninges, probably you may remember that this is a covering over the brain and spinal cord. Okay. So, this bacteria can infect those key organ, and that is quite fatal. If it is not treated, then the individual may die very quickly. Okay. And another is Haemophilus influenzae. So, Haemophilus influenzae, it's not an influenza, it's a different from influenza. So, they do share some symptom of influenza. That's why this name is there. But influenza is basically caused by virus, influenza virus. Okay, it's a Orthomyxiviridae family virus, and we will discuss when we will take up the viruses. So, all these infection we can easily treat by a simple antibiotic, but it is very important to note this infection and immediately treat, or we can treat with a wide range of antibiotics. Okay. So, with that, with this I will stop here, and in next session I will talk about other lymphoid organ such as mucosal associated lymphoid organ or lymphoid tissue. I will talk about the lymph node, and then finally I will talk about the lymphatic system, which is basically integrating all this system. Thank you very much.